glycolysis and gluconeogenesis. Glycolysis. This is a cytoplasmic pathway which breaks down glucose into 3 carbon compounds and then generates energy. As you can tell from my illustration, the pathways are in front of you of some of this cytoplasmic pathways. Glycolysis is a cytoplasmic pathway which will break down glucose into a 3 carbon compound that will generate energy. It is trapped by phosphorylation with the help of the enzyme hexokinase. Adenosine triphosphate, or ATP, is used in this reaction, and the product, which then will be glucose 6-P phosphate, which inhibits hexokinase. Glycolysis takes place in these first 10 steps, 5 of which are in the preparatory phase, and 5 are in the off phase. Step 1, hexokinase. This is the first step in glycolysis. It is the conversion of the D molecule glucose into the glucose 6-triphosphate. The enzyme that catalyzes this reaction is hexokinase. Here the glucose ring is then phosphorylated. Phosphorylation is a process of adding a phosphate group to a molecule derived from adenosine triphosphate, or ATP. As a result, at this point in which glycolysis, 1 molecule of ATP will then be consumed. The reaction occurs with the help of the enzyme hexokinase, an enzyme that catalyzes the triphosphorylation of many of these 6-member glucose ring type structures. Atomic magnesium is also involved. This helps to shield the negative charges from the phosphate group on the ATP molecule. The result of the phosphorylation then is a molecule called glucose 6-phosphate, or G6P. This is thusly called this because of the 6 carbons of the glucose that is required in the phosphorylation group, or the phosphate group. Hexokinase is an enzyme that phosphorylates a 6-carbon sugar, or hexose, to a hexophosphate. In most tissues and organisms, though, glucose is the most important substrate of hexokinases, and a glucose 6-phosphate is the most important product of these phosphorylation groups in glycolysis. Hexokinases have been found in every organism checked, ranging from everything from bacteria, yeast, plants, to humans, and even other vertebrates. They are characterized by the actin fold of the proteins, sharing a common ATP binding site core surrounded by more variable sequences that determine these substrates and other products. Several hexokinases then isoforms or isomers provide different functions that can really occur in a single species. As an example, hexokinase, or I/A, is found in all mammalian tissues and is considered a housekeeping enzyme, unaffected by most pathogenical hormones or even metabolic changes. Another example, hexokinase 2, it constitutes the principles that are regulated in this isoform in many cell types that will also increase in cancers. Hexokinase 3C, this is a substrate inhibited by glucose, and it has different concentrations. Little is known about the regulatory characteristics of this isoform. Hexokinase then, or hexokinase D, is then a cytoplasmic enzyme found in the liver and in the pancreas that basically serves to regulate glucose levels in all organisms. Glucokinase is used in the first step of most metabolism of glucose. During this step, the phosphorylation of glucose by ATP generates the glucose 6-phosphate molecule and the ADP. Glucokinase is a hexokinase enzyme. Glucokinase's role in metabolism is basically for glucose that can be inhibited due to almost all allergenic properties of glucose and glucokinase. It also plays an important role in the regulation of glucose metabolisms, and because of this regulation, glucokinase is a targeting for drug development in type 2 diabetes. Glucokinase versus other hexokinases. Glucokinase is unique from other. It has kinetic properties and is coded by different genes. The difference of glucokinase from other hexokinases is that glucokinase has a lower affinity, thus a higher Km number for glucose. The reduced affinity for glucose allows the activity then of glucokinase to differ from other conditions according to the amount of glucose that can be present. Essentially, this means that it operates only when serum glucose levels are high. Other tissues need to use glucose at lower serum levels and thus use the higher affinity or lower Km number. And glucose 6-phosphate inhibits hexokinase, and if the cell is not using the G6P that it's making, then it should stop making it, thus making the process an inhibition. In contrast, the G6P does not inhibit glucokinase and allows it to remain active and strong as much glucose as possible in the presence of high glucose levels. Step 2. Phosphoglucose isomerase. This is the second reaction in glycolysis. It is the arrangement of the glucose 6-phosphate, or G6P, into fructose 6-phosphate, or F6P. This is done by glucose phosphate isomerase. The second step of glycolysis involves the conversion of glucose 6-phosphate to this fructose 6-phosphate, or F6P. This reaction occurs with the help of an enzyme, or F6P. This reaction occurs with the help of an enzyme such as PI. And as the name of the enzyme suggests, this reaction involves isomerization reaction. The reaction involves the rearrangement of a carbon-oxygen bond to transform this 6-member ring into a 5-member ring. To rearrange or to make a rearrangement, this takes place when the 6-member ring then will open and then closes in such a way that the first carbon then becomes now the external ring, basically flipping from inside to out. PHI is an enzyme that in humans is encoded by the GPI gene on the chromosome 19,5. This gender codes a number of glucose phosphate isomerase proteins families. The encoded protein has been identified as basically a moonlighting protein based on its ability to perform this mechanism with distinct functions. In the cytoplasm, the gene product then functions as a glycometric enzyme, or glucose 6-phosphate isomerase. That then interconverts glucose 6-phosphate and fructose 6-phosphate. Extracellularly, the coded protein, also referred to as neuroleukin, functions as a neurotrophic factor that promotes the survival of the skeletal motor neurons and sensory neurons. The encoded protein is also referred to as AMF, based on additional functions as a tumor-secreted cytokine factor. Defects in the gene are the cause of most of these anemias. A severe enzyme deficiency can be such as an impairment, neurological impairment. Alternative splicing results in multiple transcript variations in this. Step 2. Step 3, phosphofructokinase. Phosphofructokinase with magnesium is basically a cofactor, and it changes your fructose 6-phosphate into fructose 1,6 biphosphate. In the third step of glycolysis, fructose 6-phosphate then is converted into fructose 1,6 biphosphate, or FBP. FBP, similar to all reactions that will occur in step 1 of glycolysis. A second molecule then of ATP provides the phosphate group that is added to the F6P molecule, the enzyme that catalyzes this reaction into PFK. As in step 1, a magnesium atom is involved to help shield negative charges in the glycopathic pathway. This pathway then catalyzes one of the key regulatory areas and has a rate-limiting steps of glycolysis by converting fructose 6-phosphate and ATP into fructose. 1,6 biphosphate has isoforms PF and PFKM, and liver PFKL4. PFKL is one of the most abundant in the liver and kidneys, whereas PFKM and PFKP are the only forms present in adult muscles and platelets, respectively. In contrast, all three isoforms are present in the brain and other tissues. 6, 7, 8. PFK1 is an inhibitor. It and ATP then will be activated by high concentrations and will break down fructose 2,6-phosphate and will add high concentrations of AMP and ADP. This is also predominant PFK1 isoforms into breast carcinoma, tumors, and B and T cell leukemias, in which total this expression of activity is if it's unregulated. However, the mechanisms underlying the regulation of PFK1 expression in cancer cells still need to be evaluated. This is step 3. Step 4, aldolase. The enzyme aldolase then splits fructose 1,6 biphosphate into two sugars that are isomers of each other. These two sugars are dihydrophosphate and glyceride 3-phosphate, or GAP as I like to call. These steps utilizes the enzyme aldolase, which catalyzes the cleavage in FBP to yield 2 3-carbon molecules. One of these molecules is called glyceride 3-phosphate, or GAP, and the other one is called dihydroxyetone phosphate, or DHAP. The enzymes then function in glycolysis to promote tumor cell growth, a cell cycle, and cancer. Isoforms of aldolase are abundant. GAP is the only molecule then that continues in this glycolytic pathway. As a result, all of the DHAP molecules produced are further acted on by this enzyme. Triphosphates isomers are TIMs, which reorganizes the DHAP into GAP, so that it can continue also in glycolysis. At this point in the cytoplasmic pathway, we have basically 3 molecules that then, but have not yet fully converted into glucose into pyruvate. Step 5. Step 6, glyceride 3-triphosphate, or what I like to call GADH. This adds an inorganic phosphate group to glyceride 3-phosphate, producing 1,3-biphosphoglycerate. In this step, two main events take place. The G3P is oxidized by a coenzyme, or NAD. The molecule then in this step, we have basically two events that will take place. We have the oxidation of the molecule in addition to a free phosphate group. The enzyme that catalyzes this reaction is glyceride 3-dehydrogenase, or GAPDH. The enzyme GAPDH contains appropriate structures then that will hold this molecule in the conformation such that it allows the NAD molecule to pull a hydrogen off the gap. Converting the NAD and the NADH, the phosphate group then attaches the GAP molecule and then will release it from the enzyme to yield the 1,3-bifoglycerate, or NADH, and a hydrogen atom. Step 6. Step 7. Phosphoglycerate kinase transfers a phosphate group from the 1,3-biphosphorates to ADP to form ATP and 3-phosphoglycerates. In this step, this enzymes will basically produce PGK. This reaction involves the loss of a phosphate group from the starting material. The phosphate is transferred then into a molecule of ADP that will yield our first molecule of ATP. Since we actually have two molecules then of 1,3-biphosphorate, because there were 3 carbon products in stage 1 and of glycolysis, we actually synthesize then 2 molecules of ATP at this step. With this synthesis of ATP, we have canceled the first 2 molecules of ATP that we used, this leaving us with a net of 0 ATP molecules up to this stage of glycolysis. Again, we see that this atom of magnesium is involved as a shield of negative charges on the phosphate groups of the ATP molecule. Step 7. Step 8. Mutase. This is where our enzyme, or our third carbon to the second carbon, to basically form the 2 phosphoglycerates. This step involves a simple rearrangement of the position of the phosphate group on the 3 phosphoglycerate molecules, making it 2-phosphoglycerate. The molecule responsible for catalyzing this reaction is called phosphoglycerate, or mutase, PGM. A mutase is an enzyme that catalyzes the transfer of a functional group from one position of a molecule to another. The reaction mechanism then will proceed by first adding an additional phosphate group to the second position of the 3-phosphoglycerate. The enzyme then removes the phosphate from the 3 position, leaving just the 2-phosphate, and then I will just yield our 2 phosphoglycerates. In this way, the enzyme is also restored to its original state. Step 8. Step 9. Enolase. The enzyme enolase removes the molecule water from the PEP. This step involves the conversion of 2-phosphoglycerates to the phosphopyruvate, or PEP. This reaction is catalyzed by the enzyme enolase. Enolase works by removing a water group or dehydrating the second phosphoglycerate. The specificity of the enzyme pocket then will allow for the reaction to occur through a series of steps that are really too complicated to cover at this part. Step 9. Step 10. Step 10, which is our kinase, pyruvate kinase. This enzyme will transfer the P from the phosphoglycerate, or PEP, to the ADP to form pyruvic acid and ATP. The result in this step is 10. The final step in 10, the final step of 10 of glycolysis converts 2 pyruvates with the help of an enzyme, pyruvate kinase. As the enzyme name suggests, this reaction involves the transfer of a phosphate group. The phosphate group then will attach to the 2 carbons of the PEP, which will then be transferred to a molecule of ADP, yielding once again ATP. Again, since there are 2 molecules of PEP, here we are actually generating 2 ATP molecules. Immediately upon finishing glycolysis, the cell must continue respiration in either an anaerobic or anaerobic direction. This choice is then based on the circumstances of a particular cell. A cell that can perform aerobic respiration and which finds itself in the presence of oxygen will continue on to the aerobic citric acid cycle in the mitochondria. If a cell then is unable to perform aerobic respiration, is in a situation where there is no oxygen, such as muscles, under extreme exertion, it will then move into a type of anaerobic respiration called fermentation. Since cells such as yeast are unable to carry out anaerobic respiration and will automatically move into a type of anaerobic respiration called alcoholic fermentation. This is step 10. Glycogenesis is a generation of glucose from a non-sugar carbon substrates like glycerol, lactate, pyruvate. The vast majority of this takes place in your liver and to a smaller extent in the cortex of the kidneys. Glycogenesis occurs during periods of fasting, starvation, or intense exercise and is highly endergonic or energy intensive. This will often is associated with ketosis. Some organs, such as the brains, the eyes, and the kidney, contain tissues that utilize glucose as their preferred or sole metabolic fuel source. This metabolic fuel source is used during prolonged fastings or vigorous exercise. Glycogen then stores and becomes depleted. The glucose then must be synthesized in order to maintain blood glucose levels. Glycolysis versus glucogenesis. Glucogenesis is essentially the reversal of glycolysis. However, to bypass the 3 highly exergonic and essential and irreversible steps of glycolysis, glucogenesis will utilize 4 unique enzymes. The enzyme unique to glycogenesis are pyruvate and carboxylase. Because of these enzymes are not present in all cell types, This can occur in specific tissues. In humans, glycogenesis takes place primarily in the liver and a lesser extent in the renal cortex. 4. Several tissues, including the brain, red blood cells, and lenses on the corona of the eye, testes, skeletal muscles during exercise, require this continuous glucose supply. Among these tissues, the brain uses glucose extensively in both the feed state and fasting state, except for this prolonged fasting, where the brain uses ketones. Notably, the daily amount of glucose used in the brain accounts for 70% of the total glucose produced by the liver in a normal fasting person. The role of allylose B. Allylose B is an enzyme for the glyconic pathways whose activity in MRA levels in the liver that'll function accordingly to diabetic status. Both the enzyme activity and the mRNA concentrate decline during fasting and increase 4 to 8-fold upon refeeding of the carbonate-rich diet. The role of the hormones and glycolytic substrates then have a gene expression in 3 tissues that will synthesize the enzyme. Fructose 1-phosphate allylase. These deficiencies are hereditary. To avoid most of this mortality associated with this condition, it must be promptly diagnosed and managed, and this activity reviews the evaluation. Then we have our regulation of phosphofructokinase, activity 1. Thank you for my presentation. My references are listed below.